Hello and welcome to the GOA-ON Webinar series. Welcome everyone, thank you for joining us for this edition of the GOA-ON Webinar series. My name is Helen Findlay and I'm here at Plymouth Marine Laboratory. I'm a biological oceanographer studying ocean acidification impacts and climate change. I'm also the Northeast Atlantic Ocean Acidification Hub Coordinator, and it's absolutely my pleasure to be moderating this panel today. This webinar series has 4 sponsoring organisations. The first is the Global Oceanification Observing Network, or GOA-ON. The second is the National Oceanic and Atmospheric Administration, or NOAA. The third is the International Atomic Energy Agency Ocean Acidification International Coordination Centre, IAEA, OA, ICC. And the final one is the International Intergovernmental Oceanographic Commission of the United Nations Educational, Scientific and Cultural Organization, the IOC UNESCO. For those of you who are new to GOA-ON, it is a collaborative international network of over 900 members from 150 countries designed to detect and understand the drivers of ocean acidification and the resulting impacts on marine ecosystems. During the presentation, all participants are in listen-only mode. You're welcome to type any questions into the question box, which can be found at the bottom of the control panel on the right-hand side of your screen. We'll be monitoring incoming questions and we will pose them to our speakers during the question and answer session, which will begin immediately after the second and final presentation. So I'd like to welcome our speakers. At this time, I will introduce Sebastein Petton first, and then Callum Hudson after Sebastian's finished his presentation. So Sebastein Petton is from the French National Institute of Ocean Science, Ifremer, in the Laboratory of Environmental Marine Sciences. Sebastian's a physical oceanographer, engineer specializing in coastal monitoring and hydrodynamic modeling. Since his arrival at Ifremer in 2012, he's maintained and developed environmental monitoring of several French sites next to oyster farms. In 2017, in close collaboration with Vilaine Oceanographic Laboratory, Sebastein implemented CFET pH sensors in the Bay of Brest. And building on this experience, he designed the French monitoring network for Carbonate thanks to the Corico CO2 project, whose objective was to obtain time series at high frequency of the pH in the coastal environment at 13 sites. And that's what Sebastein will be talking to us today. So welcome, Sebastein, and really looking forward to hearing your presentation. Thank you.
Yeah, hi, hello everyone. Thank you, Helen, for introducing me. Let me just share my screen. So now you normally should see my presentation. Is it fine? And
looks great.
yes. Yeah, okay, great. Okay, so thank you. I really appreciate the GOA-ON initiative to invite me for this webinar. It's a great opportunity for me to introduce our French coastal monitoring network for carbonate system. And as you can see on the slides, there is a lot of people involved in this network, and I also would like to thank them for their constant, continuous work and still in progress. So, as it is well known, the increase of anthropogenic carbon dioxide has induces ocean acidification. And in the same time, there is a rise of scientific and social demand due to acquire more data and define trends over the coming years. As a reminder, annual reporting of global seawater pH observation is called for under United Nations Sustainable Development Goal 14. And for several countries, monitoring the ocean acidity is now included in the water quality assessment program, as it also could be a marine environmental indicator. So my lab is specialized in studying the physiology of bivalves subjected to diverse external stresses. And it has been demonstrated that the OA process has many impacts over the different life stages of bivalve models. So it was also relevant for us to wonder what is the vulnerability of shellfish farming in such acidification process context. But before doing long-term multi-stress experiments, it is important to acquire more knowledge about the current situation in these coastal areas. This is why the Cocorico 2 project was funded for a 3-year period, and I'm going to introduce the first task, which consists in the determination of the current state of calcite and aragonite. And we really want to observe the natural variability of coastal seawater pH in this farming area. In order to doing so, we benefit from the experiments of our colleague of the Villefranche Oceanography Laboratory, who already quantified the coastal acidification in a Mediterranean site, thanks to a discrete sampling strategy with since 2007. And also they had a good experiment in high frequency monitoring since 2014. Sorry. And so in 2020, there were 4 sites which were already instrumented with high frequency probes. So the red dots here on the map are pH SAM series, and the green one are they had access to PCO2 time high series time frequency, thanks to the Roscoff laboratory team. And you may know as well that coastal monitoring can be a real challenge. So first of all, the cost of the probes can be really expensive. Secondly, depending on the geographic position of the study site, a certain amount of human resources is needed on long term to assure deployment and technical maintenance. And third, for me, is the most important. The biofouling pressure you have on site can really be important, as it could really impair your final data with regular drift. And another final constraint we had is, of course, you need to have a good expertise in the carbonate chemistry, either for operate standard procedure, do sampling strategy, lab analysis, and also data processing. So this is why nearly all the 13 monitoring sites chosen belong to already existing high frequency or low frequency program. And we decided to choose 6 different areas containing farming activities. So they're just here with a red rectangle. And in each one, a couple of sites is deployed, a more oceanic site versus one really close to oyster farm. And for another purpose of the site, I don't know if maybe you can see my cursor right now. For another project site, there is an additional one which was set in an experimental research hatchery. And at the time being, we had the choice of 2 technologies for record high frequency pH. So there was either the CFET pH from Seabird, which is based on the ISFET technologies that doesn't drift over a long time, or the SAMI pH from Sunburst Sensor, which is basically an in-situ spectrophometer. And because some of the sites of our sites are subjected to turbid water events, we decided to choose the CFET technology. So without going too much in detail, it is based on the Honeywell Giraffe sensor, and it gives 2 pH signal in total scale. But the external pH need salinity, so we needed to have an additional CTD probe deployed by them. And still, it has some drawbacks. There is no active anti-fouling system. So there is only a biofouling copper guard to put over the sensor. And the sensor requires always to be submerged. And they can have a long period of adjustment when put it in new environment. So here just some pictures of the different sites, study site. So most of the records are realized at subsurface, and we benefit from different buoys from marine channel, or of course oceanographic buoy. And 3 of the sites are actually deployed by scuba divers on metallic structures with sensor at 50 centimeters above the seabed. For the network protocol, we follow the guidelines of Bresnan and Hall method. So the CFET records data at high frequency next to autonomous CCD probes, either one from Seabird or from the French brand name NKE. And on a regularly basis, twice a month, we sample 2 bottles of half liter, according to standard procedure 1 from Dixon and al. And the first one is for pH measurement with a spectrophotometric method. And the other one is for the measurement of alkalinity and dissolved inorganic carbon, which is going to be measured by the French National Lab SNAPO CO2. For the spectrophotometric measurement, we really want to achieve an accuracy of the order of the thousands of pH, as we want to be able to correct the C-fade raw signal in case of drift. So for that, we gave an initial training, and we should organize also different annual intercomparison exercise for all the laboratory involved in the project. So to conclude, so this method for the methodology, so it's the same methodology is applied to every site for sampling, for data collection, and for data processing with Python. And concerning uncertainties, we want to reach out the weather objective for high frequency data and the climate objective for low frequency data. So as the different data sets are required, it is important to assess their reliability. For the discrete sampling, we first do some sanity check. For example, we can compare spectrophotometric pH with an estimation of pH based on alkalinity and DIC. And even if there is some uncertainty in the numerical estimation, it is a quick way to remove outliers due to bad storage, air contamination, or even measurement error. And thanks to the discrete sample, we can now validate the raw pH signal and correct the C-fade coefficient if needed. So here on the upper graph, you get the two raw pH signals, and the black dots represent the spectrophotometric sample. And below, you get the difference between the correct pH signal and the discrete sample. So we can now select the best fitted pH signal for deployment period. And basically, our main result right now is nearly 2-year cycle of pH monitoring in each areas. So on this graph, the blue signals is the oceanic site, and the red signals are the farming site, and the line is just the daily average pH. So here just an example for 3 particular area, and we can clearly see the difference between each of them. The scale here is the same for each of them. And there is also the different variation between the oceanic and farming site. So really quickly, the top one is an oceanic bay with low freshwater runoff. The second one is the Arcachon Basin, is really the largest basin for oyster recruitment in France. And the difference of pH between both sites is mainly due to salinity effect. And for the last one, it's quite particular because the farming site is located inside the Tow Lagoon, whereas the oceanic site is situated outside in the Mediterranean Sea. And the next step right now is to determine the whole carbonate system. So to do so, we use a linear relationship between discrete alkalinity and salinity. So it fits well in most cases, even if there is a lack of data for low salinity value. And then with the PyCO2sys package, we can have access to all the parameters we want for each area for both oceanic and farming sites. Here, just a quick example with PCO2 and saturation state of aragonite for the Northern Atlantic site. Honestly, we still have to work on it to maybe find a better regression for high frequency alkalinity, and maybe by including tide cycle or nutrient data. And I think this requires specific crews in each region. And the final goal we have is we can start to characterize the monthly distribution of aragonite saturation states and to compare oceanic and farming sites. So here's the code color is the same. So farming site in red and blue, oceanic site in blue. And you get the monthly distribution of aragonite for each area. And the pie chart here just represents the percentage of observation under a certain threshold. And I just put here a 1.5 limit for an example. And I think this is typically the kind of risk indicator you can propose to policymakers. So we can see that depending on the areas, the risk of lower aragonite availability is not happening in the same time of the year. So it's mainly for winter for Atlantic sites. And for the Tour Lagoon, it's quite particular because in August, September, and October, this is where we get low pH value, and this could be quite alarming, and as it could be have a dramatic impact on recruitment success with lower values. And if we look more now in detail on the data, we can easily determine biological cycle. So here on the upper graph, you get a scatter plot of the high frequency pH. The color corresponds to the hour of the day, and the black line is the daily average pH. So we can see that the green points, which are the evening pH, are higher. So the pH is higher in the evening and lower in the morning. And this is mainly due to respiration and photosynthesis processes. But this is not particularly true in spring here, where we don't see this kind of pattern. But now if we look at the daily variation, so it's the bottom plot, we can see some kind of tilt shape according to time. And this is typically the sign of a semi-diurnal tidal cycle in this area. So there must be a bloom in the river, which goes back and forth due to the ebb and the flood tide. And we can also have a look to, it is also possible to determine the direct relation between the pH and also parameters we can measure on the data. So for example, oxygen and fluorescence data. So here you have the data from the Mollet Bureau. It's a bureau located in the Bay of Vilaine. It's an open bay towards the Atlantic Ocean. And there are two particular events. There were two particular events last summer. If we look at the first event in April 2022, we got a sudden drop of salinity, and this drop of salinity put much more freshwater input, and there was a bloom of nanoplankton, which we can be seen on the fluorescence data. And these data are quite correlated with the pH and the oxygen as well. And a bit later, in June, we get another bloom, but it's dinoflagellate. And what is quite interesting is after the bloom degradation, we can see a massive drop of pH directly correlated with the oxygen drop as well. So it's just really interesting to have such signal to determine the complexity of the biological processes. Nevertheless, you may have noticed some gaps in the data, and this is mainly due to biofouling. So fortnightly manual cleaning is not sufficient in some times. So we tried different solutions. The first one, we use a sea fox. The sea fox from Seabird is an SB37 clamped to the sea fats. And it uses the TBT anti-fouling devices to protect the sea fat sensors. But as you can see on the picture, after just 3 weeks in the low turbidity water, the whole system clogged. So it wasn't really good for us. We also tried different solutions, actually not recommended by Seabird. So we tried a UV lamp, but it was definitely a fail. As the ISFAT is really sensitive to it, but we are quite happy right now with the chlorination pumping system. And this is just a test we did last summer by deploying 2 CFAT on the same buoy. The red one is the CFAT in the standalone mode, and the blue one is equipped with the chlorination system developed by Ifremer. And there was no manual cleaning for the CFAT with the chlorination during the 2 months of deployment. And we can see that there is no heat, just we had some kind of, we do manual cleaning for the CFAT in standalone mode, but we can see that there is no drift in the CFAT with chlorination. So it's really encouraging but we need to evaluate the long-term impact of chlorination system on the ESFET. And for now, it's still going on, and it's quite good. So we are quite happy with that. And of course, sometimes we can wonder what type of pH we are measuring on our buoys. So of course, when the sensors are completely full by microorganisms, like this one, the picture just after 15 days in the Arcachon basin, we know that the data are wrong. But sometimes it's a complete probe itself, which is surrounded by mussels. So we are more measuring their physiology and their respiration rate rather than their environmental evolution. And despite these difficulties, we need to follow our efforts and to acquire long-term pH series, as it could be valuable for plenty of subjects, I think, such as lab experiments, satellite-derived products, or for coastal modeling. And we are right now actually waiting for another call of the European Maritime Fisheries Funds to support this network. And we're also including two more stations in the Mediterranean Sea. And we have in mind to test another sensor, maybe less precise, but that does not require in situ calibration. So we are still trying to test it. We are still waiting for it, actually. And we want to also to develop other technical solutions to reduce maintenance and manual cleaning. And another part of this project is also to add special crews, especially to better characterize the relation between alkalinity and salinity in the sites. And also, as it was a requirement of our policymaker to transfer this method and to tell them how it is done in the film. So I thank you for your attention. And if you have any question, I'd be glad to answer it. Thank you.
That's great. Thanks so much, Sebastian. Really interesting talk, and great to see how the data is coming together. We'll take questions at the end after Callum's talk, so please remember to put any questions in the Q&A box at the bottom of the screen. And we will now move on to Callum and come back to those questions shortly. Thanks, Sebastian. So Callum Hudson, thank you for joining us. Callum is our next speaker. He's currently a PhD student at Okinawa Institute of Ocean Science and Technology Graduate University in the Marine Climate Change Unit. Callum completed his Master's for Research at the University of Plymouth here in the UK with Dr. Ben Harvey, also at the University of Tsukuba, I probably said that wrong, apologies, to study the impacts of oceanification and typhoons on algal communities at natural CO2 seep sites. He's now a first-year PhD student in Okinawa, Japan, supervised by Dr. Harvey and also Professor Tim Ravison. Callum is looking at CO2 sites and other natural analogues to study the impacts of global change in marine communities and the impacts of oceanification and warming of the ability of urchins to exert top-down control and prevent transitions to turf states. So another really interesting talk about bringing kind of the natural analog side of things into understanding the impacts of oceanification. So Callum, I'll hand over to you and go take it away. Thanks very much.
Sure, cool, thank you. I'll just try and get my screen up. How's that? Good? Perfect. Cool, awesome. Cool, thanks Helen for the introduction. Yep, I'm currently in Japan, Okinawa, and I'm going to be working with Dr. Ben Harvey for my PhD mainly. But this project is something I did for my master's, sadly not in Japan, but whilst I was sat at home in Plymouth for the year during COVID. But I'm really grateful that Ben let me kind of use this data for my thesis, and I had a good time playing around with it and kind of looking at this site and system, and I look forward to like doing more with it in the future. So if I get started, see if I can. Cool. So I won't go over kind of some of the basics of OA, but what I really do want to kind of touch upon is how this duality of ocean acidification as both a resource for some organisms and a stressor for others has really important implications for its ability to reconfigure marine communities, raising concerns around the potential for regime shifts in marine systems, whereby these communities can undergo drastic, abrupt, and persistent changes in their composition, structure, and functioning, And as a result, ecosystems can shift to new baselines or alternative stable states, often resulting in their simplification and the reduction of diversity and structural complexity. And this is potentially particularly worrying when regime shifts are discontinuous or non-linear, meaning that they display hysteresis. And so kind of a simple reversal of conditions in the environment is insufficient to return that ecosystem back to its pre-disturbance or pre-tipping point state. And so one of these major transitions that we're increasingly observing in the marine environment is this replacement of macroalgal or kelp forests by these turfed algal states, whereby the reef kind of changes from a complex forest to a structurally simple mat of low-lying algae. And these shifts towards turfs represent a widespread global loss of habitat structure and this move away from traditional algal structuring dynamics of urchin grazing and overfishing towards kind of more climate and nutrient driven dynamics. And so some previous work at some of these different CO2 seeps, such as the one that we used in this research, has shown that once these turf algae become established, often at high CO2, high kind of future OA conditions, they displace this coral and macroalgae. And then once established, they kind of become locked in further by different stabilizing feedback mechanisms, which reinforce their dominance and lock the degraded state into place. So for example, macroalgal recruitment is reduced with greater turf cover due to kind of physical inhibition, but also the way that these turfs trap sediment and then also change the kind of bacterial community structure within and below the turf, creating this kind of anoxic layer, which is harmful for new macroalgal or coral recruits. The turf are also boosted in their growth and production under high CO2, and the kind of a loss of herbivores and grazing pressure can also help to promote their dominance. But some of the mechanisms through which these transitions can occur due to acidification and whether additional environmental drivers are involved, we thought required more study. And so in this project, we tried to look at how physical disturbance from typhoons, which is this seasonal pulse stressor, how it impacts algal communities at different levels of acidification. So typhoons, I don't know if all of you know, I've kind of got a bit used to them now living in Japan for the last year or so. They're very powerful weather systems characterized by strong winds and heavy rainfall. And like OA, they also have the potential to restructure benthic communities. And so because of this, we questioned whether there might exist some potential for the effects of OA and typhoon disturbance to interact together. So as I kind of mentioned briefly, in Japan, typhoons are a highly seasonal pulse stressor, generally peaking in their frequency towards the late summer and the early autumn when sea surface temperatures have risen to their kind of annual highs. And many of these storms form in the tropics, and then they kind of track northwards, making landfall on the mainland of Japan, having passed over and disturbed coastal areas in the south, such as where our study site was located for this research. So just quickly in this study, we aim to assess how changes in the structure of algal communities driven by ocean acidification mediates their stability against periods of physical disturbance by typhoons. This seasonal pulse stressor, which comes through kind of periodically to exert kind of large impact on the algal communities. So we wanted to look at how these communities were able to resist damage during storms, but also how they could then subsequently recover after periods of disturbance. So natural ecosystems are structured by both physical processes, but also the interactions between coexisting species within these ecosystems. Therefore, although very practical and very useful, we got a lot of information from them. Lab-based studies looking at single or few species can sometimes overlook some of these effects. And so in this project, like many other kind of research going on all over the world, we examined algal communities at a well-studied CO2 seep, this one being the vent at Shikanejima, which is kind of an island located off the coast of Japan's main island. And these shallow water volcanic CO2 seeps, such as Shikanejima, create natural gradients of CO2, offering insights into future oceanic conditions through space for time analyses. And these provide the potential for OA studies to consider both community and ecosystem level impacts by studying the responses of multiple species and whole ecosystems and communities to these naturally fluctuating environments. Shikanejima itself is a warm temperate system dominated by large stands of macroalgae. and it also often falls beneath the path of many typhoons as they track northwards. And so this study, they were kind of regularly and with each year kind of largely impacted by these typhoons, and that's why we kind of wanted to look into this a bit further. So in this study at Shikanejima, we compared the algal communities at 3 locations around the seep site. So firstly, we looked at this reference area outside of the influence of the CO2 seep, and we used this as like a control for modern day conditions. Secondly, we used a near future elevated CO2 area, and lastly, an end of the century high CO2 area. And I'll refer to these from now on as reference elevated and high respectively. So to look at the effects of ocean acidification, but also these seasonal differences in algal community structure throughout the year and between years, we performed monthly surveys over a 3-year period, photographing permanent quadrats bolted onto the substrate at each of these sites, the reference elevated and high CO2 location. And then we take these photos and overlay an 8x8 grid of points onto each photo quadrat. And then I'd go in and image J, kind of zoom in, look at what algal group, what kind of algae was under each of those points, and assign a functional group to that point. And so each community, for example, these photos on the left and right, we'd get 64 data points of different functional groups, which would form a single kind of community at a different time period and at a different CO2 location. We also then assigned scores, so 0 to 5, to each of the different functional groups that we used in this study, based on the relative amount of habitat structure that they provide, so that we could calculate and sort of estimate the structural complexity of these different communities. So for example, a community comprised of high biomass of macrophytes, such as the photo on the left here, had a relatively high structural complexity, whereas the community on the right, which is dominated by a large mat of these low-lying turfs, was relatively less complex. And as we can see from these photos already here, we get a sense of the clear differences that exist between the reference and high CO2 communities as we expected. So indeed, Permanova analysis showed that these algal community compositions did differ between locations, so they changed with increasing OA. And to kind of visualize these changes throughout time a little more easily, we made these stacked bar plots to compare the mean relative abundance of different algal functional groups within communities between months, so through time, and also at these different levels of CO2. So we saw this kind of clear cycling in the composition of communities and the dominance of different functional groups over time and between seasons. And this also differed at different levels of acidification. So for example, we see these spikes of turf algal cover, particularly during the summer months, as well as more bare rock during the autumn and winter at the acidified sites in particular. Whereas on the other hand, at the reference sites, there was a larger amount of structurally complex macrophytes, as well as more calcareous algae, with the highest macroalgal biomass occurring in the spring and summer, much like the turf for the acidified sites. And when we performed pairwise comparisons of these community compositions at different CO2 levels within each individual month, we kind of saw and confirmed that the differences in composition were still significant and maintained within and between those months. So for example, for 9 of the 12 months of the year, the composition of the communities at the elevated and high CO2 sites did not significantly differ from one another. It's just suggesting that they were kind of changing and responding to environmental change in similar ways, but they each themselves differed significantly from the reference or modern day communities, which were associated with different functional groups. So more macrophytes, foliose algae, CCA and other calcareous forms of algae. So kind of having shown and seen that algal communities differ both with increasing acidification, but also kind of between seasons and how this differs, we next examined in more detail how these differences alter the impact of typhoons when they come through during those periods of typhoon disturbance, by looking at changes in algal cover, structural complexity, and community composition, both before and after these periods of extreme disturbance by the typhoons. So again, already from the photos here, we get an idea of the representative seascapes at the different CO2 sites before and after typhoons come through. And we get a good idea of the pattern that occurs. So the algal cover is reduced, but to a greater extent at the high CO2 sites, which essentially completely reset the community, reducing it to bare rock by ripping away these matrix-like mats of turf algae which come to dominate during the early summer at these high CO2 sites. So to look at that in more detail, if we first focus on changes in the algal coverage, we saw that whilst the communities at each site display similar levels of peak levels of algal coverage, so during these yellow recovery and blue pre-disturbance, pre-typhoon season periods, directly after and during the typhoon season, this algal cover was reduced, and this was more pronounced in the acidified communities. Also, the coverage of the reference communities, they seem to remain more stable over time as they displayed more resistance against the typhoon disturbance. And despite this, despite exhibiting lower resistance, the acidified communities still were able to rapidly recover following the typhoon season. Again, in this yellow and blue recovery and pre-disturbance periods, coming back to this near pre-disturbance level of cover, recovering to high levels of algal coverage. We then made these generalized linear mixed effect models using quadrat, month, and year as nested random effects to test the impacts of timing, so whether the communities were before, after, or in this recovery period, as well as CO2 condition on the algal community cover and complexity. And this GLMM found for coverage at least that timing, but not CO2, had a significant effect on algal coverage. And when we compared these algal coverages between each of these time periods with post-hoc pairwise permanovas, we confirmed that the algal communities under different CO2 conditions responded to typhoon disturbance differently. So we can see here that algal percentage cover before and after typhoons differed significantly at each location, being reduced during the typhoon season once that disturbance comes through. Now, for the reference sites, algal cover returned rapidly. So there's no significant difference between these before and recovery communities. However, this was not the case for the acidified communities, perhaps due to this larger impact of the typhoons as they come through and rip away the algal communities that are living there. So now if we go back and focus on the changes in structural complexity of these different communities, we saw that the acidified communities generally possessed a lower structural complexity, and this was again reduced further during this typhoon season. Now, whilst this complexity does appear to recover after typhoon disturbance, it only does so to pre-disturbance baselines, which again are lower than that of the reference communities, which maintain that higher level of complexity through time generally. The structural complexity of algal communities was also significantly impacted by both typhoon disturbance and CO2 level, and at all sites, structural complexity differed significantly before and after the typhoons, again being reduced by that disturbance. But again, using these pairwise analyses, we saw that for all levels of acidification, the complexity before and in this recovery period following typhoons, the communities either side of the typhoon peak season do not differ in their complexity. So they are able to recover to this pre-disturbance level of complexity. But this is they do not recover in the same ways, and I'll go on to talk about this a little bit more, but rather in ways that are shaped by the differences in the dominant groups at each of those sites. So here we use principal component analysis to plot the composition of the algal communities at these different levels of CO2, and comparing how they changed and differ before and after this disturbance by typhoons to look further and more closely into how the community composition at the sites explains these differences in percentage cover and structural complexity. So from these figures, we see that the acidification-driven differences in community composition and the varied responses of the different algal functional groups to typhoon disturbance might explain the changes that we saw. So these, again, pairwise permanovas show that typhoons had a large impact on algal community composition, again, differing before versus after typhoons. There's a significant shift whereby the community changes in response to disturbance. And this seemed to be particularly the case for the acidified communities, once again, which significantly differ for all pairs of timings, so not just before and after typhoons, but also these before and recovery communities also significantly differ in their composition, so they're not able to fully recover. Despite being impacted by typhoons, the composition of the algal communities at the reference CO2 levels seemed to be more stable, so they were able to return to their pre-disturbance composition by this recovery period, despite being changed directly after disturbance. So to bring some of these ideas together, generally the ecosystem state of present-day communities was higher than those under future conditions, in part due to this persistent press disturbance from ocean acidification, which leads to changes in community composition, lower functional diversity, and reduced structural complexity. And these acidification-driven differences in community composition, and as well as the associated differences in the functional traits and life histories of different species and the functional groups, led to reductions in ecosystem stability in the face of disturbance, particularly for the future turf-dominated communities, but less so for the modern macroalgal-dominated communities. So these macroalgal assemblages appeared more resistant to disturbance from typhoons, maintaining this more consistent composition between seasons and recovering to pre-disturbance baselines following physical disturbance by typhoons. Whereas the turf-dominated communities displayed lower resistance to typhoons, which almost entirely removed the algal community, reducing that community to basically bare substrate ready for succession to begin again. When it did, turf resilience, promoted by boosted primary production and growth under these elevated CO2 conditions, meant that the empty space created by typhoons was then recolonized by turf algae, not macroalgae. And this then also likely prevented later macroalgal recruitment due to those feedback mechanisms that I mentioned earlier. And we suggest that regular disturbance at these sites, due to the seasonal disturbance from typhoons, helps to lock in and maintain this turf-dominated state by maintaining this early successional stage within which turf can rapidly grow and recolonize and come to dominate and lock out other more complex algal groups. Cool, so that's all from me. Yeah, I guess it's question time if there's any.
That's great. Thanks so much, Callum. Really interesting to hear about that work and to see the complexities in that environment that we maybe don't capture so well in sort of lab studies. So really nice to be able to bring lab and field work together. So we do have some questions, so I'm going to go straight into those. And I think we'll start with a couple for Sebastian first. So we have one from Frederick Gazoo. Why do you think diurnal variation is higher with chlorination, Sebastian?
Yeah, so this actually was a bit of a trick because the system we tested, it was not a final system. The chlorination, we derived the flux of the chlorination directly to the CFAT, but without controlling the synchronization of the CFAT measurement and the chlorination. And of course, we know that there is an effect of 0. Depending on the electrode, there is an effect of the chlorination on the SFAT. And we cannot for now control it. And this is what we have to do right now is to realize an automatic controller that control plus the CFAT measurement and also the chlorination system. And in this graph I show, there was just a bit of a mistake because there is some measurements which are influenced by the chlorination. So this is why you see a higher diurnal variation, but it's not true.
Brilliant, thanks. We're going to pop one to Callum. There's also one from Frederick Gazoo. What is the variability of the pH or PCO2 at the sites, if you can, Callum?
I was just scrambling to try and find it because I can't remember.
I can give you some more time if you.
Yeah,
that
would be great. I can answer
yeah.
the next one though, if that's.
Yeah, so Jeremy Carlot said, "Hi Callum, thanks for the great presentation. I know you've mentioned it, but I missed it. What is complexity and how did you define it?"
So here we were looking at structural or habitat complexity of the communities. So we took that kind of 8x8 grid, assigned a functional group to each of those to get that kind of assemblage, and then the different functional groups. So for example, like turf or CCA, bare rock, were assigned a score from 1 to 5, depending on how much structure they provide. So like bare rock was 0, and a kind of a, I don't know, like a complex macrophyte was like 5 or something. And so we basically added those all up and divided it by the total potential score, which was what, 64 times 5, so 320, I guess. And then we normalized it, I believe. Yeah, so kind of habitat complexity, structural complexity of the kind of whole community. Yeah.
Sounds good. Hope that helped clear that up. So we'll jump back to Sebastian. We've got a couple more questions for you. Manuel Correa, I probably said that wrong, I apologize. The effect of pH change is more noticeable in mollusks than in fish. Is it possible to know how pH changes could influence the distribution of fishes or their physiology, based on kind of the work that you're involved with, I guess?
For me, it's a bit out of my scope because I'm really a physical oceanographer, and it's quite hard to answer that question. But I think, yeah, it's there are people working on it, and yeah. But I can't answer really that question for me. Sorry.
Yeah, I think that's the fish question is quite a complex one, and there's a lot of debate in the community, I think. But it's always nice to try and be able to correlate some of this real world data that we have and understand what's going on, especially when it comes to kind of modeling. So this modeling work that looks at how fish distributions might change with warming, and people are starting to now think about how we can introduce the impacts of acidification on that. But that's for another day, I think. So Sebastian, we have another question from Christophe Rabuel. The relationship of salinity and alkalinity shows very different patterns in the different basins. Could you explain why?
Yeah, so this is quite surprising at first, because we have kind of either the alkalinity is really increasing with the salinity, or it's completely the opposite. And this is actually part of the distribution of the composition of the sediment, and what is in the river actually, if there is some kind of limestone or such things. And the Atlantic sites, all of the sites are showing linear increasing relationship, except for the Marano-Leron site. And indeed, in that case, there is a really, there is a lot of limestones in the sediments. And so the freshwater runoff brings more alkalinity. So it's quite the opposite. And for the Mediterranean site, of course, the Mediterranean is really has a lot of high alkalinity rate. So we see this difference, and we can now, I don't know how to explain this relation for the Mediterranean side, but I will have to look at for it. And this is part of the next job we want to do in the next project.
Yeah, that's great. I actually was going to ask a similar question related to the alkalinity relationship, because we are monitoring sites in the UK. They're a little bit further offshore than I think some of yours are. So I was just interested to see actually that some of your sites, most of them are set on the bottom, the sensors, or sort of 50 centimeters or so off the bottom, I think you said.
Actually, yeah.
Yeah, so that's interesting. What sort of water depth are they in then? Is it just a couple of meters of water, or does it depend?
Yeah, it depends. Actually, most of them are deployed at subsurface, so yeah, 2-3 meters below surfaces, and yeah, but only 3 of them at 15 centimeters. So yeah, but I think it's really depending on the situation, the geographic situation in the estuaries, which decided if there is a lot of low freshwater runoff or not. This is a good case.
Yeah, that's true. And because we get quite a lot of organic alkalinity coming out of the rivers near us, so I guess that's probably another factor to think about as part of all of that. But really interesting, and it'd be great to see that funding come to fruition, where you can go and do some more analysis of that with some cruises. So that's great. So if we pop back, Callum, do you have any more information about the variability of pH and PCO2 for your sites, or do you want to come back? Maybe you could chat with Fred offline about that.
I was muted, but I've got the paper up here, and so it was using CFET measurements over roughly a month. We had the reference location pH of 8.137 plus or minus 0.056, and at the highest, we had 7.788 with plus or minus 0.106 standard deviation. Yeah.
Okay, so
it's in the paper. If I can,
it's in the paper.
Yeah, there's a I just couldn't remember off the top of my head.
That's cool. It's interesting. I was going to actually. One of my questions was, how does the typhoons actually influence the water chemistry? It would be quite interesting to see if there's much disturbance of that in terms of pulling along different water properties for a certain period of time, or whether that affects the gas bubbling that comes out of these CO2 vents or anything. I don't know if you've observed any of that. But
I haven't because I haven't actually been to the CO2 seep site yet. I did it all from my bedroom in Cornwall when I was during COVID. But I guess we don't know and we haven't looked at that. But with the kind of pretty large scale of these typhoons, as they rip through, I'm sure they do kind of churn up the water quite a lot and mix things up. But kind of, I guess the main focus that we looked on here was that physical impact rather than that kind of chemical changing in pH. Yeah. Also, if you put the expensive sensors out during a typhoon, you're probably going to lose a few, and then this
is one of the problems, isn't it, we have with these sort of measurements, is that you don't want to lose your instruments, but actually capturing that sort of information is pretty tricky. So looking at remote sensing options, perhaps, of how we can get around these sort of issues as oceanographers and things. But yeah, things for future note and future work. So that's great. So we have an interesting question from Lionel Feiger. Apologies again if I get that wrong. Greetings from the Solomon Islands. I really like both your presentations. Since you're working to capture global data, how can you build your networks to the South Pacific? So I guess this is a little less specific for both Callum and Sebastian, but maybe you can have a think about whether there's opportunities as a graduate in the Solomon Islands. Leonor says he has concern related to the CO difference in different parts of the world. So if anyone's got some thoughts on that, how we can help to build capacity in other areas, or any advice that you would provide to these sort of people that are just graduating and thinking about setting up networks or setting up monitoring. Maybe Sebastian, you have a few comments about the challenges you've experienced or recommendations you can make.
Yes, as I told you, yes, price. Actually, this is why I just made the slide about the coastal monitoring challenges. And I think for me, it's always the thing for it to work is it's really hard to build it from nowhere. If there is nothing there, it's really hard, because there is a lot of to get a certain amount of data and quality data. You need to have access to a lot of means, or either oceanographic mooring, data laboratory for chemistry analysis, and of course the different probes. So I think the GOA-ON furnished some kind of WebKit for global acidification observing. I'm pretty sure of that. So this could be a good start. And afterward, if you want to do high frequency, you really have to be sure that you can access to the different sites really quickly due to biofilling pressure. And this is
I can just add to that, and I think the Secretariat are probably busy typing away or reaching out to you. There is a South Pacific hub. Well, there's several hubs in that region. There's the South Asian hub, which has recently started, and there's also the Pacific Islands and Territories regional hub, and they're doing quite a lot of work on capacity building in the South Pacific and that kind of area. So it may be worth just reaching out to the Secretariat to find out who to contact for that, or having a look on the GOA-ON website, which is goon.org, I believe. I'm sure the Secretariat can correct me if I'm wrong on that. And also, so there's also the GOA-ON in a box kit, which Sebastian mentioned, which is basically a set of equipment that is relatively cheap comparatively compared to some of the other stuff, but should kind of get you up and running with lots of kind of visuals and capacity building research and things. Not research, that's not the right word. My brain is failing me this morning. I'm full of cold. just some guidance, resources, that was the word I was looking for, resources available on the website as well. So I think we've got time for this last quick question from Urias Taylor, which I think is to Sebastian again, which was, what was the objective or rationale for selecting specific locations for pH monitoring?
So yes, so as you see, before 2020, we only had 4 sites with carbonate high frequency monitoring, and we really want to have more data to describe the variability of pH. And as you can see with the data, there is a lot of variability between the areas, and inside each area, between the oceanic and the farming side, there is also different variability. So I think it's really important to describe this variability, maybe for 5 years, just to know how it is, we wonder, because we didn't know for sure how it was, or it was a long, maybe in the past there was a kind of such monitoring was done with a former pH sensor with glass electrodes, which were not really reliable. So this is why we choose a lot of sites just to describe the different variability. And I think we were right, because there was a lot of variability between each of them.
Great, thank you. So I think we better end there. It's coming round to the on the hour mark. So I'd like to just one last time say thank you very much to our speakers, Callum and Sebastian. It's really great for the to hear their presentations and really interesting work that's being done, and hopefully lots of takeaways there. If you need any more information, please just get in touch with Secretariat, and we can always put you through to Callum and Sebastian if you have more direct questions. I'd also like to thank you, the audience, for attending and joining us for this edition of the webinar series. And if you'd like to stay up to date with the GOAN community, consider sign up as a GOAN member. Sarah's just put the web link in the chat there, but it's www.goon.org/members/join. We'll be announcing the next webinar on Twitter and our GOAN email listserv, so please watch out for those. And thank you all again, and hopefully see you all next time. Wonderful.
Thank you.